It's a yeah. It's a real pleasure to have with us Professor Byron Yu from Biomedical Engineering and Electrical and Computer Engineering at CMU. Byron did his graduate school at Stanford in Krishna Shenoy's lab, and then working with monkeys, and then with computational models or computational work, recording from multiple neurons. And then he did a postdoc at UCL, the Gatsby Institute, with Manish Sahni, and then moved to CMU, where he has been for a long time in the engineering departments, multiple ones of them. And he has been working on multiple areas of work, but primarily focusing on methods to deal with data sets coming from recordings from multiple neurons at the same time and implementing them and using them for brain-computer interfaces. So it's the latter part that he will talk to us about today. So all over to you, Byron.
All right, so thanks a lot for the kind invitation, Suresh, and all of you for coming. So just by way of introduction, I thought I'd tell you a little bit about my research group and what we work on. So broadly speaking, we seek to understand how large populations of neurons process information and carry out computations from sensory systems to motor systems and also what's in between. And we're a computational group, and we collaborate very closely with experimental groups in various places. And as Suresh mentioned, during my PhD, I did experimental work for the first half of my PhD. And that has, at the time, I didn't really appreciate it. It felt, like very difficult and very, very constraining, but I've realized in hindsight that I wouldn't give up that experience for anything else. And so, being a computational person, I feel like I'm better able to understand experimental constraints and data more deeply and also collaborate more effectively. Okay, so the work in the group has a few categories of projects. So one is brain-computer interfaces for addressing basic scientific questions about brain function. And this is together with Aaron Batista at University of Pittsburgh and Stephen Chase at CMU. The second category of projects involves studying how populations of neurons in different brain areas interact with each other. And as you probably know, think of any brain function that we use on a daily basis, whether it be any sensory function or decision making or any motor function. It involves interactions between brain areas, and our understanding of how populations of neurons interact to enable these functions is still sort of at its infancy. And so we've been working together with Adam Cohn at Einstein College of Medicine, Christian Machens at Champalimaud, and also in separate projects with Matt Smith at CMU to understand these interactions between brain areas and how they underlie various brain functions. The third category is developing and applying statistical methods for studying neural population activity, involving dimensionality reduction, dynamical systems, et cetera. Okay, so today I'm going to focus on the first of the three categories. Okay, so before I dive in, I just want to acknowledge the people who led this work. Matt Golub, he was a PhD student with us. He just started as an assistant professor at University of Washington. Emily Obi is a postdoc with us. She is on the job market right now. She wants to lead a non-human primate lab. And Patrick Sadler was a PhD student with us, now is in industry. Jay Hennig was a PhD student with us, is now a postdoc at Harvard. And all of them were co-advised by Professors Aaron Batista, Steve Chase, and me. Okay, So when you hear the term brain-computer interface, the first thing you probably think about is a paralyzed person who is controlling a robotic limb. Okay, so this is a very important use of brain-computer interfaces. But there's another use of brain-computer interfaces which has been less recognized and which I think is perhaps as important. And this is the use of a brain-computer interface as an experimental tool to understand various brain functions. And so this is sort of the topic of today's talk. Okay, so the central message for today is that a brain-computer interface is a powerful tool for basic science. Okay, and why might this be? Well, a brain-computer interface simplifies the output interface of the brain without hopefully simplifying away the complexities of brain processing that we seek to understand. Okay, so for example, let's take arm reaching. Okay, so the output interface, or the effector here, is the arm. Okay, the number of non-output neurons, this is all the neurons in your brain, essentially, are in the many millions. Okay? The number of output neurons, okay, say these are the neurons that project down the spinal cord, right, are in the many thousands, right? And in any given experiment, only a small number of them are recorded at any given time. And oftentimes we don't even know which neurons we're recording from are actually the ones projecting down the spinal cord or not. Okay, furthermore, the causal mapping between neural activity and arm movements is typically unknown, right? Usually studies find a correlative relationship between the neural activity and movements, but not a causal mapping. Furthermore, the effector dynamics, in this case, the joints of the arm and the muscles, right? They are nonlinear, right? And difficult to measure. So some heroic studies have gone about defining these nonlinear dynamical equations to define all the joints in the arm, but most studies don't do that. Okay, and furthermore, sensory feedback, and in particular here, I'm talking about proprioceptive feedback, is tied to the arm. Okay, so the output interface for arm reaching is rather complex. And so what we propose is to simplify matters using a brain-computer interface. Okay, and a brain-computer interface is a system by which you record neural activity from many neurons in the brain, and then you translate that neural activity into movements of a cursor on a computer screen using a so-called BCI mapping. Okay, and this mapping can take the following form, okay, where here xt is a 2x1 vector. Okay, that is the horizontal velocity and vertical velocity of the computer cursor. Okay, xt minus 1 is the velocity at the previous time point. U sub t here is the neural activity. Okay, so if we're recording from 100 neurons, ut is a 100-dimensional vector where each element represents the number of spikes, right, or the activity level of that neuron at that time step. Okay, and then a, b, and little b are parameters of this BCI mapping, which we as experimenters can choose. Okay, and so the subject's task is to change its neural activity so as to move a computer cursor to hit visual targets under moment-by-moment visual feedback. Okay, feel free to ask questions if you have any throughout. Okay, so now let's come back to this table, right, and ask for BCI. Okay, for this as the output interface. Now we have the effector is now a computer cursor, right, or a robotic limb. Okay, here the non-output neurons are still in the many millions, right? All the neurons in the brain can still contribute to moving a cursor to hit targets on a screen. Right? However, a key difference here is the number of output neurons here is by definition, those neurons that we recorded, right? So if we recorded 100 neurons, right, those are the 100 neurons that eventually, whose activity will move the computer cursor. All the other neurons in the brain can still participate as long as they feed their activity through the neurons that we record. Okay, so this is going to be extremely powerful because we get to record the entire set of control signals, the entire set of output signals, right, in the system. And so we see any changes in behavior, right, changes in due to learning, for example, we can trace those changes back to the activity of one or more of the neurons that we recorded. Okay, so here the neuron to movement mapping is known because we define what those matrices are, A, B, and little b. Okay, and the effector dynamics can be linear, right? So the equation that we just saw was a linear equation, and that's going to help us define various hypotheses rigorously and be able to test them, okay, experimentally. Okay, and also sensory feedback, in this case, proprioceptive feedback, is not linked to where the cursor is, but linked to the arm, right? So you can imagine designing experiments where you dissociate where the cursor goes with where the proprioceptive feedback goes, and to ask questions about proprioception. Okay, other people have done very interesting experiments. We're not going to talk about that today. Okay, so I'm going to show you a video of a monkey controlling a computer cursor using a BCI. And this is representative of current state of the art, right? So this is with 20-ish neurons recorded from primary motor cortex. Okay, and the monkey is moving the screen cursor in the center to hit the blue targets presented on the screen. Okay, so you see that these movements are quite fast and quite accurate. Okay, so movements are typically around 1 second, and accuracy is typically about 100%. Okay, and so by most metrics, this is about half as good as what we can do with our arms. Okay, and the reason why I'm showing this to you is because we're going to be using BCI to address scientific questions. And a prerequisite to doing that is that we have good behavioral control. Right, and so I hope, from this video, you can see that the starting point is good behavioral control. Okay, so there are several key advantages of using BCI for asking scientific questions. Okay, so the first is that we can monitor all neurons that directly drive movement, right? And so as I mentioned a moment ago, this will allow us to make scientifically causal statements about the relationships between changes in neural activity and changes in behavior. Okay? And usually in studies of arm movements or eye movements, we do not have that causal relationship, but rather a correlative relationship. Okay, secondly, we can distinguish between output and non-output neurons. Okay, so this is relevant to questions, for example, relating to, say, interactions between brain areas, right, where where only a fraction of the neurons in any given brain area project to other areas, right? And so you can imagine setting up a BCI where you record from, say, 100 neurons, you use 50 of the neurons to drive movement, but then you watch or record the activity of the other 50 neurons passively to see how they change their activity. The third is that we can define a simple mapping, okay, like the one we saw earlier. And that allows us to define scientific hypotheses rigorously and test them rigorously in data. Okay, fourth, we can change the mapping. Okay, so we can change those parameters, A, B, and little b, to create a learning experiment, right? Where the animal will now need to figure out how to use this new BCI. Okay, and then fifthly, we can flexibly manipulate the proprioceptive feedback. Okay, so what we're going to talk about today, we're going to leverage advantages 1, 3, and 4. Okay. So we're going to talk today about learning, okay, and how a BCI allows us or enables us to study learning. And learning requires the brain to alter the neural activity it produces. Okay, so for example, if you're learning to play tennis or you're learning to manipulate a Rubik's Cube like a pro, right? Something changes in your brain between when you're a novice to when you become an expert. Okay, so we sought out to ask two fundamental questions. Okay, so one is why are some tasks easier to learn than others? And then the second is how does neural population activity change during learning? Okay, so why are some tasks easier to learn than others? Well, different tasks require the brain to produce different neural activity patterns. And so if it's easier for your brain to learn or to produce certain activity patterns than others, right? This might be a reason why certain tasks may be easier to perform than others. And can we go as far as to ask, can the brain produce arbitrary activity patterns? And if not, what defines these constraints on learning? Okay, so here is the setup, or here's how we think about the population activity. Okay, so this is a cartoon where we're recording from 3 neurons, right? In the actual experiments, we're recording more like 100 neurons. Okay, and so we can then define this population activity space where each axis represents the activity level of one of the neurons we're recording. Okay, so we're recording from 100 neurons. This would be an 100-dimensional space. So then what we can do is to take a window and count the number of spikes that each neuron produces within that window. That defines a point in this space. We can do this for many such time bins, and we get a scatter of points, okay, in this space. Now, we can define the BCI mapping in this space. Okay, so this is a graphical representation of what is defined by the big A, big B, and little b. Okay, so now, if this is the BCI mapping, if the subject wants to move the cursor to the left, the subject would need to produce neural activity down here. Okay, whereas if the subject wanted to move the cursor to the right, they would need to produce neural activity over here. Okay, so now the key experimental manipulation here is that we change the BCI mapping unannounced to the subject. Okay, like so. Okay, and now the subject needs to learn to use this new mapping through trial and error. Okay, so now under this new mapping, if the subject wants to move the cursor to the left, it would need to produce neural activity up here. And similarly, if it wanted to move the cursor to the right, it would now need to produce neural activity down here. Okay. So when we started this experiment, we did a few different instances of this. And we found that for certain new mappings, the animal is able to learn to use it really quickly. And with other new mappings, the animal had a real hard time at learning to use a new mapping. And that led us to ask whether there was some principle underlying which new mappings would be easier to learn and which new mappings would be more difficult to learn. Okay, so this led us to propose a hypothesis, okay, that is based on an empirical observation that we and many others in the field have made in different brain areas, different species, different tasks, which is that neuropopulation activity tends to not to populate this entire population activity space equally. Okay, but rather, neural activity tends to live close to some lower dimensional space within the higher dimensional population activity space, okay, which we're going to call the intrinsic manifold. Okay, so for those of you, who are mathematically inclined or oriented. Here, when you hear the word manifold, you might think of nonlinear spaces, okay? But so in general, this could be a nonlinear space, and we can talk and think about it in terms of nonlinear space. But for the experiments that we're going to talk about, we're going to identify a linear manifold from the recorded activity. Okay. So the hypothesis goes as follows, okay, that if we start with a mapping like so, right, this is a mapping that the animal can use really well, like what you saw in the video. Okay, now, if we switch this BCI mapping so that the new mapping remains within the intrinsic manifold, that this new mapping would be easier to learn. However, if the new mapping is outside of the intrinsic manifold, that this new mapping would be more difficult to learn. Okay, so at this moment, some of you might be saying, wait a minute, like, isn't this obvious? Like, why would you even spend your time doing this experiment? Okay, so let me put two things on the table. Okay, the first thing is that it is not the case that right after we switch the mapping, that this within manifold mapping is easy and this outside manifold mapping is difficult. That would not be an interesting experiment. Okay, instead, a really important control in this experiment was that we needed to ensure that right after we switched the mapping, that the initial difficulty of each of these two types of mappings was not different from each other. And then from that point, we want to see how learning proceeds. Okay, the second thing to put on the table is that one might think at first that these outside manifold mappings are impossible. Like how could it be the case that a subject could learn to do this? Okay, it turns out that there were multiple studies that were done before us, which did these sort of outside manifold mappings and showed that animals were able to learn them under certain circumstances. Okay? And they didn't call them outside manifold mapping, but if you look at the methods and how they switch the mappings, they were of the outside manifold variety. Okay, so this is to say that it's not the case that these outside manifold mappings are impossible for animals to learn. Okay, everyone okay with this so far? Yeah, okay. Okay, so now let me just tell you a little bit more about the specifics of how we did this experiment. Okay, so the standard place to start for anyone who wants to do BCI decoding is to use a Kalman filter to convert neural activity into cursor or arm movements. Okay, so for the purposes of this experiment, we inserted factor analysis as a pre-processing stage on the neural activity to allow us to disambiguate between these two different types of switched mappings. Okay, where here we start off with neural activity, which is around 90 dimensional, so around 90 neurons. Okay. Factor analysis then identified a 10-dimensional intrinsic manifold from the recorded neural activity. Okay, and this number 10 was determined from the data using cross-validation. Okay, and then these 10 factors were then passed into the Kalman filter to obtain a cursor velocity, which then moved the cursor on the screen. Okay, so this is what we had in the experiments: 90, 10, 2. And then in the illustrations, we had 3, 2, 1. Okay, so now to define within manifold perturbation, what we did was we took these 10 factors and we permuted them before we input them into the Kalman filter. Okay, and what this served to do was to keep the relationship between the neurons and the factors the same. Okay, but what this changed was the relationship between each of the factors and cursor movement. Okay, so in other words, to learn to use a within manifold perturbation, the correlation patterns, right, amongst the neurons didn't need to change, right? The subject only needed to produce the right activity patterns at the right time. Okay, in contrast, for the outside manifold perturbations, what we did was we took the neurons and permuted them before inputting them into factor analysis. Okay, so now for the animal to be able to learn an outside manifold perturbation, what it would need to do is to produce new correlation patterns amongst the neurons so that they would be suitable for moving the cursor to hit the targets. Okay, so a few experimental methods here. So our experiments were used Utah array recordings. So these are arrays of electrodes with a 10 by 10 electrode arrangement in primary motor cortex of macaque monkeys. We used threshold crossings, okay, so no spike sorting. Spike counts were taken in non-overlapping 45 millisecond bins. Okay, and the animal did an 8-target center out task, okay, to move the cursor from the center to each of 8 possible targets presented on the screen. Yeah, Eric.
Can you tell me just briefly where that 45 millisecond window came from?
Yeah, why is it 45 and not? Yeah. So this is has now turned into an inside joke in the group. Okay, which is, so the student who was who led this work, he was playing around with different window sizes and empirically trying to assess, oh, should we use a 30 millisecond window, a 60 millisecond window, right? And then asking how the animal was able to control the cursor with each of those. And neither of them was particularly great. And so he said, okay, why don't we split the difference and try 45? And then it seemed to work better than 30 or 60. And so we just went with that. And by better, I just mean the basic control before we introduce any of these perturbations. And so now all the experiments are using 45 millisecond windows because of this one historical artifact. But if you use 30, if you use any window, you know, that's a few tens of milliseconds, I think the performance of moving the cursor would be quite similar.
What about wider windows? Can you still
wider?
100?
Yeah. So then there starts to be a trade-off if you get up to 100 or 200, right? Which is that then the updates, right, between successive cursor movements, right, the time steps are longer. And so then the subject's going to feel more of a delay in, you know, I produce neural activity. When do I see the effect of that neural activity show up on the screen?
The window reduces the variability in your spike.
That's right, also.
Right? And it does that in a nonlinear fashion. Your window is on variance, right?
Yeah,
down.
exactly.
I was wondering if you guys explore trade-offs between longer windows, more accuracy in estimating the neural activity versus a delay versus small.
Yeah. So in an earlier paper, when I was a graduate student, we explored that. So broadly speaking, if you choose a window between 20 milliseconds and 100 milliseconds, the performance is pretty comparable, although you can optimize within there. But you don't want to go too much larger than 100 or too much smaller than 20 because of the effects that you just described. Okay, so let me show you the results from a couple of representative sessions of these experiments. Okay, so on the left here, this is an example of a within manifold perturbation session. Okay, so on the axis here, the vertical axis here is the animal's success rate, okay, of hitting the targets. Horizontal axis here is the trial count throughout the day. Okay, so the whole horizontal axis here is about an hour and a half of experimental time. Okay, and one trial is moving the cursor from the center to hit a target. Okay, so at the very beginning, we see that the animal performs at 100%. Okay, just like what you saw in the video. And then unannounced to the animal, this vertical dashed line, we switch the mapping on the animal. And so as you would expect, the performance drops. Okay. And through trial and error, the animal can then learn to use this new mapping. And here the animal comes back to close to 100%. Okay. And at the end of the experiment, we then have the animal work with the first mapping again. Okay, and so what you see here is a lot of learning. Okay. Okay, now contrast that with what we see on the right, which is a representative session for one of these outside manifold perturbations. Okay, so here, again, at the beginning of the session, the animal is essentially 100% correct. Okay, and then we switch the mapping. And so as you would expect, performance goes down. Okay, and here we see maybe a little learning, but then it peters out, right? And then at the end of the experiment, we show the first mapping again. Okay, so 1 second. Okay, so here we see less learning than here. Yeah.
Do I understand correctly that when you're perturbing this manifold perturbation, it's just completely random, right? So there is a possibility that this new vector has some sort of projection onto your linear manifold.
Yes. So in all of these cases, the new mapping has a substantial projection. Okay. Has substantial similarity with the original mapping. Okay. I should say. Okay. And the reason is the following. So, okay. So for either of these two different types of perturbations, there are millions of possibilities of which we need to choose one to conduct in any given experimental session. How do we choose that one? Okay, so what we did was we enumerated, for in this case, all of the possibilities, all of the 10 factorial possibilities, and we enumerated millions of these possibilities at the beginning of the session. And for each of those candidate perturbations, we predicted how initially easy or difficult it would be for the animal. And the reason why it was important to do this prediction was that if right after we switched the mapping, the animal would perform with 100% correct with that, then there's no learning to be had. And so that would not be an interesting experiment. Okay. Conversely, if right after we switched the mapping, the animal's performance was essentially 0%, the animal would give up and not try to learn anymore. And so what we tried to do was to choose a new mapping so that it had intermediate difficulty, where there was still something to learn. At the same time, it wouldn't frustrate the animal so much, it would give up. Yeah, and across all the different sessions, we had a distribution of these initial difficulties, right? Here and here. And those were the two distributions that we needed to ensure were matched between the two types of perturbation. Yeah.
So just a quick question. Can you remind me how do you obtain the outside manifold perturbation? Do you just shuffle the spike kind of the neurons?
Yeah, so we shuffle the identities of the neurons. So
you have 19 neurons in your 14.
Yeah.
So you want to shuffle through these neurons. So what does it mean?
Yeah. So one way to think about it is like two neurons that used to be positively correlated would now need to show negative correlations.
So you just change the weight of the weight of like all of these neurons.
Exactly. That's one way of thinking about it. Yeah. So that the velocity of the cursor is some linear combination of the activity of all the neurons. Right? And each of these types of perturbations changes the weights of that linear combination in some specified way.
So that's what you, by changing D, you're changing outside manifold perturbation, right?
By changing B, okay, you can think about the outside manifold perturbation as, okay, so B is a 2 by 90. If you have 90 neurons, right? B is a 2 by 90 matrix. And so an outside manifold perturbation is, you can think about it as permuting the columns of B, which is mathematically equivalent to permuting the elements of, permuting the neurons.
If you obtain perturbations by just permutations either of neurons or of factors, how do you determine the parameters of the Kalman filter, which I assume do not change when you do these perturbations?
Sorry, I didn't understand. One more time.
You also have parameters inside your Kalman filter, right?
Yeah.
And how do you determine these? Because they're fixed across perturbations.
Yes.
So you must have chosen them in order to ensure that learning can happen sufficiently quickly. So how do you do that?
Yeah. Okay. So at the very beginning of the experiment, we have the animal passively watch the cursor moving on the screen. And it turns out that the neurons in motor cortex change their activity in accordance with the direction in which the cursor is moving. Okay, this might sound very surprising to you because we're in motor cortex, not visual cortex, right? And it turns out, so the way in which I think about how this could be the case is that, the animal has already done, this type of task, right? Where it's moving its arm or it's using a BCI to move a cursor to hit a target. And so even if there is a series of trials where the animal is not actually in control of the cursor, is still thinking about, okay, I'm going to push the cursor to try to hit the target because it knows that that's the goal. And so as soon as the animal thinks that, then that sort of volitional activity shows up in the motor cortex. And so that is what we leverage at the very beginning of the experiment for just a few minutes to then find the parameters of the Kalman filter, which are then fixed, as you say, which we use for the remainder of the experiment.
That makes sense. Thank you.
Yeah. Okay, so we did tens of sessions of each of these types of perturbations across multiple animals. Okay, and the consistent effect that we saw was that we saw more learning in this case than in this case. Okay, and that's the main result. Okay, so just to summarize the first part, so we see more learning within the intrinsic manifold than outside the intrinsic manifold. Okay, and this implies that the existing structure of a network can shape learning. Okay, because the intrinsic manifold represents or arises from constraints imposed by the underlying network in terms of what the activity correlations are amongst the neurons. A third point here is that people in the field, including us, have used dimensionality reduction to visualize data, right? And people have asked, can dimensionality reduction do anything beyond data visualization? Okay, and so here's an example in which we used dimensionality reduction as part of the experiment, right, to reveal constraints on what the brain is capable of achieving. Okay. So now let's move on to ask the second question, which is, how does the neural population activity change during learning? Okay, so what we're going to do is to focus on these within manifold perturbation sessions, because these are the sessions in which we saw the most learning. Okay, and what we're going to do is to now look into or on to the intrinsic manifold and ask, how does the neural activity reorganize during the learning process? Okay, so here we're now looking onto the intrinsic manifold. Okay, and here is our starting point when the animal is controlling the cursor very well, like what you saw in the video. Okay, and so this is a cartoon where for this initial BCI mapping, when the animal wants to move the cursor to the left, it produces these green points. When the animal wants to move the cursor to the right, it produces these purple points. Okay, if it does that, the animal will perform very well with this mapping, okay, like what you saw in the video. Okay, now we switch the mapping, right, unannounced to the animal. Okay, and our key question for the second part of the talk here is how do these clouds of points move around during the learning process? Okay, so what we did was we defined 3 hypotheses of what could happen. Okay, and then we asked which of the 3 hypotheses was most consistent with the data. Okay, so the first hypothesis is called realignment. Okay, and you can think about this hypothesis as changing the neural activity in a way that maximizes the behavioral performance under the new mapping. Okay, so what you see here, according to this hypothesis, the green cloud, which was here before, would migrate up here to be consistent with this new mapping of moving the cursor to the left. And this purple mapping, which used to be here, migrated down here to be consistent with this new mapping for moving the cursor to the right. Okay, so before we started this project, I would have put a lot of money that this hypothesis would win. Okay, so we also tested 2 other hypotheses. The second hypothesis is called rescaling. Okay, and you can think about this as restoring the influence of neural activity on behavior. Okay, so meaning that previously, changes in neural activity, remember that the original mapping was like this, right? Okay, so previously, under that mapping, changes in neural activity along the horizontal direction here would have big changes on where the cursor would go. Okay, and now under this new mapping, changes in neural activity in the horizontal direction have less of an influence. If you think about the projection, right, of neural activity onto now this line, right? Okay, so to restore that influence of the neural activity, the variance of the neural activity, according to this hypothesis, would scale up or down along certain dimensions such that the variance of the neural activity when projected onto the mapping was preserved. Okay, and this is an analogy to visual motor gain adaptation studies that come before us. Okay, the third hypothesis is called reassociation. Okay, and you can think about this hypothesis as repurposing existing activity patterns. Okay, whereby in this cartoon, I kept all the points the same. All I did was recolor the points. Okay, so we have this so-called neural repertoire here, according to this hypothesis, which is hard to violate. But within the neural repertoire, the animal can choose to decide when to use which activity pattern appropriate for the task. And so the ones that are more up here would be produced when the animal wants to move the cursor to the left, and the neural activity patterns down here would be produced when the animal wants to move the cursor to the right. Okay, so we did various tests. We did various visualizations to begin with, and then we did various quantitative tests to ask which of the hypotheses was most consistent with the data. Okay, and without taking you through all of that gory detail, you can I welcome you to look at the paper, but let me just tell you the punchline, okay, which was in all of those tests, hypothesis 3 was the clear winner. So what this implies, right? If we think across the results from part 1 and also now this part, is that neural activity seems to be not only constrained to this low dimensional space, i.e. the intrinsic manifold, but within the intrinsic manifold, it seems that there are further constraints where neural activity cannot go anywhere within the intrinsic manifold, but rather there is some neural repertoire, some space of activity patterns, right, which tend to be easier for the animal to learn to produce. And the learning happens within that repertoire rather than going to new parts of this intrinsic manifold. Okay, so that was the first bullet here. Second bullet is that because, so hypothesis 1 is optimizing behavioral performance, maximizing behavioral performance. Okay. Hypothesis 3 does not maximize behavioral performance. Okay, but that is most consistent with the data. Okay, and so what this suggests is that learning is suboptimal on the time scale of hours, the duration of our experiments, in that it doesn't maximize performance. We've also done this experiment over multiple days, where rather than giving the animal 1 to 2 hours to learn a new mapping, we allow the animal to use the same mapping again and again and again across 2 weeks to see if the animal can show more learning. And it turns out that the animal can. Okay, even with these outside manifold perturbations. Okay, and it seems that both of these types of constraints, both intrinsic manifold constraint as well as the neural repertoire constraint, can be overcome to some extent if you give the subject more time. Okay, so now let's sort of hop up a level and come back to the very beginning of this talk, where we talked about how BCI is a tool for doing basic science. Okay, and so the insights that we've obtained here were made possible by putting the task requirements directly on the neurons, okay, rather than indirectly based on arm or eye movements, right? Which is more typical of experiments that we conduct in the field. Okay, and because we put these task requirements directly on the neurons, and because we're able to record from the complete set of control signals used to determine behavior, the cursor movements, we can causally attribute behavioral changes, i.e. in this case learning, to specific changes in the recorded activity. Okay, and this causal point, I think, is a powerful one. Okay, and it was enabled by these hypotheses in the second part that we defined, were enabled by the fact that we defined this mapping between neuron and neural activity and movements to be linear. Okay, of course, we could have defined a nonlinear mapping, but that would have made these hypotheses more difficult to test or adjudicate between. Okay. We've also used BCIs to study several other questions involving internal models of sensorimotor control. Okay, so the punchline of this story is that the animal is not perfect, when using these mappings, even the ones, like you saw in the video, sometimes the cursor doesn't move perfectly straight to the target. Okay, and we asked why this was the case. And it turns out that most of the errors that the animal makes can be attributed to the animal using some internal model of the BCI mapping that was not a perfect match with the actual BCI mapping that it was using, because we never told the animal what the mapping was. The animal needs to figure it out from trial and error. And what it figured out was something that approximated the truth, but wasn't exactly the truth. And that could explain a lot of the behavioral errors that the animal is making. Okay, we've also studied the question of neural redundancy, okay, which is there are lots of possible activity patterns that the brain can produce. Okay, it's a really high dimensional space because there are so many neurons in the brain. Okay, the number of degrees of freedom of our movement is much smaller. Right, and so that implies that there are many possible activity patterns that the brain can produce to lead to the same movement. Okay, then amongst all those activity patterns that can produce the same movement, which of those activity patterns does a subject actually produce? Right, that was the question of this study. Okay, and then in addition, we've used BCIs to ask how internal states, okay, such as arousal and motivation, are interwoven with the learning process. Okay, and it turns out that in the same experiments that we talked about today, there are these huge changes in neural activity that happen, especially at the beginning of the learning process, that appears to have nothing to do with learning, but rather has something to do with arousal. Okay, and because the signal is correlated with pupil size. Okay, and it turns out that that change in the neural activity has a substantial influence on the whole learning process and what plays out after that. Okay, and then in addition, more recently, we've been studying BCIs. We've been studying the concept of memory, okay, in using BCIs. Okay, so when you learn something, right, you want to be able to remember it, right? And it turns out that when we have the animal use the initial mapping and then a perturbed mapping, and then we have the animal use the first mapping again, that in principle, the neural activity that the animal produces in this in task A block, the animal can produce the same exact neural activity as it produced here during the first task A block, right? But it doesn't. It turns out that the animal produces, the neural activity that the animal produces here during the second task A block is different from the neural activity that the animal produced in this block in a specific way, okay, that is specific to what the animal learned in the intervening time. Okay, and so in other words, the animal is able to do task A proficiently while retaining a memory of what it learned during this task B block. Okay, furthermore, we've been comparing biological learning with machine learning, okay, where in machine learning, machine learning is all about optimization, right? You have some system, you set up an optimization, an objective, right? And then you perform an optimization to get the machine to learn. Okay, and the question is, can we think about biological learning as an optimization? Okay, and it turns out there are various observations about how the brain learns, okay, that we've stumbled across and others in the field have looked at as well, okay, that currently are not part of standard machine learning models. Okay, and so we posed the question in this perspectives piece of whether, incorporating some of those concepts that we've discovered about biological learning might lead to better machine learners. Okay, and now hopping up yet another level. Okay, so these studies that we talked about today, as well as others in the field which are using BCI, what we're seeing is that BCIs simplify the brain's output interface without hopefully simplifying the complexity of brain processing. Okay, and these studies that we and others have done are showing that they engage many of the same cognitive processes as we use during arm reaching and eye movements. Okay, so concepts such as learning, internal models, and more. Okay, and then hopefully BCIs can be used to study other sensory motor and cognitive processes, right, outside of the motor cortex. Okay, and so these are people in my group and funding sources. Yeah, happy to take questions.
Great. Okay, Byron, thank you so much. There were two questions during the talk from.
Should I turn off?
I think I should just.
Yeah, I think that would be.
Yeah, there were two questions that came up during the talk. One was from Vivian. She wanted to know if the responses of motor cortical neurons when watching the cursors move could be related to mirror neurons.
Yeah. So yeah, could be. So. Yeah, I think this is an interesting topic of further study, and other people have shown, right, in motor cortex and other places, these effects. So yes.
Yeah, and the second question was from Puya. He was asking whether hypothesis 3, the winning hypothesis, does that mean that learning only happens downstream from the recording area?
Okay, so this is very interesting. So is maybe Puya can maybe say, yeah, maybe. Okay, so let me tell you the way in which we've been thinking about it, and then you can say more about how what your thoughts are, if that's okay. So we've been thinking that for these within manifold perturbations, what the animal is doing is changing the inputs to the motor cortex, right? So you imagine, we're recording from the motor cortex. Motor cortex is inputs from, other brain areas, right? Premotor cortex, parietal cortex, etc. Right? And now, if all you do is to change the inputs to motor cortex, right, to tell it which activity patterns to produce when, right, that would be consistent with effects that we found, right, which is that the neural activity remains within some intrinsic manifold and also remains within the neural repertoire. Okay, whereas for the animal to be able to use one of these outside manifold perturbations, what it would need to do is to rewire the local circuit in M1 in some sense to show new correlation patterns, where two neurons which were positively correlated may now need to negatively correlate. And that takes more time, it's more difficult. Okay, so so just to summarize, we've been thinking about it in terms of changes in inputs to M1 versus changes in the local connectivity within M1. I'm intrigued by your question because I think in your question, you said that this might imply that the learning happens downstream of M1. Do you have some thoughts about that?
You guys hear me?
Yeah.
Yeah, I think everything you said, it fully rhymed with what I was thinking. I asked the question and like a second later, I realized that in your setup, there's no downstream because you're just reading out of the motor cortex. But I think what I was thinking was an area like prefrontal cortex that I think you also mentioned. It's like some process like gating happening. And as you said, like as an outcome, the input to the motor cortex would change without actually the circuits being changed, right? But maybe now that I have the opportunity to just ask the question, a follow-up question would be, how much of these findings would you expect to really depend on the setup that you're using to learn, to study learning? In a sense, I'm feeling like your setup is turning motor cortex into a limb, so that the animal, like internally, it's like, I'm trying to imagine what the animal is experiencing. And it kind of feels like the animal is trying to learn how to move its new limb, which is like a bunch of neurons, right? And you're forcing it, in a sense, to a set of movements that are within the manifold. It's kind of like, for me, it's like, if I know how to move my right arm, I'm being asked to move it in a different pattern, for example. And how much of this learning would you expect to be different from if the animal is asked to be doing something like playing the piano, which the animal has never done? It requires coordination between like two hands now, something that maybe the animal hasn't done before. Then in that case, would you say that you expect something to change in the local circuit of motor cortex, and learning would, as a result, would have a different characteristic? Just curious to hear what your thoughts are.
Direction what you're thinking, right? Because ultimately, what we want to understand, is, learning during, everyday learning, right? We're, taking dance class or a sports class or learning a new language, right? So we're really viewing this as a stepping stone, right, towards that, right? Where the ultimate goal is to understand, how learning and some of these other concepts play in to sort of more natural tasks. And maybe some of the concepts that we've, been able to study using a BCI, maybe now we can apply those concepts to these, maybe less simple settings.
All right, thanks a lot.
Really interesting stuff. I always like it when I hear about this, read about it. But some of the details you put at the initial of your talk made me think about it. Just tell me if I'm thinking about this right. Your cursor position, when you use your BCI to map neural activity of cursor position, relies on the previous cursor position. That's part of the.
Yes.
That doesn't have to be in that term, right?
That's right.
You could take that term off, and my neural activity can put that cursor anywhere, at any
That's
time,
right.
in any way.
That's right.
Did including that term in your map of the previous cursor location force the manifold? Have you ever tried it without it? Like I can just do a magical map between neural activity and put my cursor anywhere I want on the screen instantaneously.
Yeah. So two parts of makes me think of two things. Okay, so the first thing is if you don't have that a times xt minus 1 term in there, the cursor will be really jumpy.
Yeah, which is okay.
Yeah. I mean, it makes it a bit harder for the animal to. I mean, the animal's already very experienced. They'll probably tolerate that just fine. Okay. But if the animal is sort of new to this whole setting, they'll find that to be a frustrating experience. And it turns out that that A matrix is almost the identity matrix, but has its diagonal values a little bit smaller than 1. Okay, so what you could think about this is doing is saying that the velocity at this time step is almost exactly the same as the velocity at the previous time step, with some small change induced by the neural activity at this time point. So the contribution of the velocity from the previous time step is quite big, which allows us to have a smoothly moving cursor. Okay, but you're right. We could do the experiment, without it, and there may be ways to design the experiment so the animal would not be frustrated. In terms of, whether that could create the manifold, one way of thinking about this is maybe outside the context of BCI, but in the context of arm movements, right? In the very same neurons, right? We can record from the same neurons, have the animal perform arm movements, right? And what we see is that the neural activity does not uniformly populate the entire population activity space, but rather some subset of that space. And then there are interesting questions like, okay, well, is the low dimensional space populated by the neural activity during arm movements similar or different from the neural activity populated by the neural activity during BCI movements? Okay, it turns out. So we're studying that question right now. There are some similarities, there are some differences, right?
I had one final question, actually related to your answer to Puya's. So I don't know where the UJARA was recording from. Presumably it was the arm neurons. What I'm thinking of is, what is the movement that corresponds to the new spaces occupied by the neurons in the naturals, when the monkey's naturally doing it? In other words, this is related to what the monkey might be doing to reorganize it, right? So, for example, in motor adaptation experiments, when you have the cursor adapting, so instead of moving this way, you move this way to move the cursor. So clearly you have these two patterns, they've occupied new, you've labeled them so that you have two new spaces. Yeah. But what is the movement that corresponds to the new labels, which might indicate what the monkey might have been aiming for in order to do the new remap task? Right?
Yes.
What would the monkey have to think to create these two labels?
Yes.
Which would be its strategy to do the new task?
Yes, right? Yes. So I love the direction in which you're thinking. We haven't fully clarified this, but we could start asking these questions in the data. Okay, so the interesting thing here is that, okay, the animal is doing a task where it's getting two-dimensional visual feedback, right? It's looking at a screen, right? Yet, the space in which like the perturbations are happening and where its neural activity lives is in a 10-dimensional space, right? And so in some of these behavioral experiments that you're referring to, Suresh, the perturbations are defined within the two-dimensional behavioral space. Like, for example, a visual motor rotation, right, is a rotation within the two-dimensional space. Okay. But here, what we have is a visual motor rotation or something like it within the 10-dimensional space, of which the animal sees two of the dimensions, right? And so, some of these perturbations might look to the animal like a visual motor perturbation within the two-dimensional space, but it's not going to be exactly that because the perturbation was defined in the 10D space. Yeah. So exactly as you said, like we could, so what we're trying to understand now is, okay, in more detail, like what is okay, so in the second part of the talk, what we compared was before learning versus after learning. right? And so what we really want to understand is how did it get from the before learning stage to the pre-learning to the post-learning neural activity. And then, through this process, we want to know like what were the ways in which these sort of associations were changed during the learning process.
Fantastic. So we are running out of time. So thank you very much, Byron. And thanks everybody on Zoom for coming. The 56 people on Zoom. So that's a good number. Great. Thank you very much.